Okay, let's unpack this. Have you ever, stopped to consider the invisible world teeming right beneath our feet? I mean, this hidden realm of microscopic life, stuff that can just devastate our crops, spread nasty plant viruses, and even hunt down tiny worms with what's basically a biological gun. Today, we're taking a deep dive into exactly that kind of hidden universe. We're exploring a really fascinating and honestly sometimes a bit unsettling group, the Plasmodiophoromycota and their intriguing cousins, Haptoglossa.
Yeah, and what's truly remarkable, I think, is how our understanding has shifted. For a long time, they were studied by mycologists, the scientists who focus on fungi. But modern DNA analysis, well, it now places them squarely within the protozoa. That's a completely different branch on the tree of life. So we're going to explore their pretty unique biology, their often bizarre life cycles, and the surprisingly sophisticated ways they infect their hosts. We'll try and paint a vivid picture for you, all without any visuals.
Right, so our mission in this deep dive is to give you a clear, engaging, and hopefully vivid understanding of these really important but often overlooked players in our ecosystems. They have a huge impact, especially, in agriculture and biotechnology. So get ready to imagine a world where a microscopic amoeba can cause a massive root club and a single-celled organism can actually fire a biological bullet. Meet the Plasmodiophoromycota, the unseen agricultural saboteurs.
Okay, so if we look at the bigger picture, these organisms, they really had a bit of an identity crisis for ages. For decades, mycologists, scientists studying fungi, they included them in their work. But, like I said, sophisticated DNA sequencing, it's shown they aren't related to true fungi, or even the mycota, the water molds. No, instead, they seem to be distant relatives of slime molds and definitely belong with the protozoa.
Huh. So they're like those distant, slightly odd relatives you just found out about at the family reunion, with a really surprising backstory. But despite being tiny, I think you said only about 12 genera and 51 known species? They pack a huge punch. How do we even tell these minuscule things apart?
Well, primarily, scientists differentiate the genera based on how their resting spores are arranged inside the host cell. It sounds technical, but it's key. For instance, in Polymyxa, you'll see lots of resting spores clustered together. It forms a neat group called a sorus. But then you look at Spongospora, and the resting spores are grouped more loosely. It looks almost like a tiny sponge, hence the name.
Okay, and the impact they have, that's where it gets really significant, right? They aren't just like scientific curiosities, they're obligate parasites, which means they absolutely need a host to survive. And some of their favorite hosts happen to be our most economically important crops.
Exactly. They cause major plant diseases that can just devastate yields. Think of clubroot disease and things like cabbage and broccoli, that's Plasmodiophora brassicae, or powdery scab on potatoes, that's Spongospora subterranea, and even crickroot disease in watercress from Spongospora nasturtii.
So what does this actually mean for, our food supply,
Hmm.
for agriculture globally?
Well, beyond the direct damage, which is bad enough, some species also act as silent carriers. Vectors for important plant viruses. Spongospora subterranea, for example, it transmits the potato mop-top virus. That can severely cut down tuber yield. Then you have Polymyxa beta carrying beet necrotic yellow vein virus, and P. graminease carrying several mosaic viruses affecting major cereals like wheat and barley. And the real challenge here, these viruses can hang around for years, hidden inside the pathogen spores in the soil, even when you're not growing the host crops.
Wow, so they're like tiny Trojan horses waiting in the soil.
Yeah.
A deep dive into Plasmodiophorales, life cycle and infection strategies. Okay, let's zoom in a bit. What do these tiny invaders actually look like? How do they get around?
Right, so their mobile stage, the infective stage, is called a zoospore. Picture these microscopic swimmers. They have two whip-like tails, or flagella, but they're unequal in length. One's shorter than the other. Imagine a tiny swimmer with two uneven propellers kind of thrashing its way through water films in the soil.
And once they find a suitable plant root, how do they actually get inside and set up shop?
Okay, once they're inside the host cell, they transform. Their main body becomes this wall-less amoeba, and this amoeba grows into something called a plasmodium. You should picture a blob of cytoplasm, just full of nuclei, moving freely inside the plant cell. This plasmodium is amoeboid, meaning it can stick out these false feet, pseudopodia, and it can actually engulf parts of the host cytoplasm. It's basically cell-eating, phagocytosis. This is thought to be a very ancient, kind of primitive trait, maybe hinting that their ancestors were free-living amoebae.
A blob-like invader that eats parts of its host from the inside. That's pretty insidious. Can it spread further once it's in one cell?
Oh, absolutely. These amoeboid plasmodia, they're quite mobile within the plant tissue. They can actually digest their way right through the plant cell walls, moving from an infected cell to the next healthy one. So the infection spreads throughout the root system. But while this internal plasmodium is wall-less, other stages do have walls, like the little cysts the zoospores form on the root surface, or the tough resting spores that help them survive in the soil for so long.
Okay, with these sort of remarkable entry methods, let's trace the whole journey. How do they reproduce and spread? What's the full life cycle look like?
Right. It typically kicks off with one of those tough resting spores, which, as we said, can just sit dormant in the soil for years. Many years. When conditions are right moisture, maybe signals from a host root, the spore germinates. It releases a single primary zoospore. This zoospore swims around until it finds a good spot on a plant surface, like a root hair. Then it settles down, secretes a cell wall around itself, and encysts. It's like deploying a tiny self-contained landing pod.
Okay, the pod has landed. What happens next? How does it breach the defenses?
So from this cyst, a wall-less amoeba is injected, quite literally injected into the host cell. This amoeba then grows, forms that plasmodium we talked about. And here's a really unique feature of this group. Its nuclei divide in this very unusual way called cruciform division. Under a microscope, the nucleolus looks like it elongates into a cross shape during division. It's quite distinctive. This first plasmodium, the primary one, then makes more zoospores, secondary zoospores. These get released and can reinfect the host nearby, leading to this rapid asexual cycle of spread, multiplying quickly. But eventually, a different type of plasmodium forms. This one undergoes genetic recombination, meiosis, and produces those new, hardy resting spores, completing the whole cycle. Though the exact points where sexual fusion happens, that's still actually a bit debated by scientists.
Okay, so that's the complex biology. What does this actually look like in a plant? Let's take Plasmodiophora brassicae, the clubroot one you mentioned.
Right, in infected crucifers, cabbage, cauliflower, turnips, that family, you'll see these greatly swollen, gnarled roots. Both the main taproot and the side roots can get affected. Sometimes they look a bit like swollen fingers or toes. That's where the old name finger and toe disease comes from. Above ground, the symptoms might be subtle at first, maybe just wilting leaves on a warm day, but then they strangely recover overnight. Later, though, the plants become stunted, they turn yellowish, and their growth just slows right down.
Oh.
Seedlings can even be killed outright. And this massive swelling, it's caused by two things happening together. Hypertrophy, where individual host cells get bigger, and hyperplasia, where the host cells divide more than they should. It's uncontrolled growth.
That sounds absolutely devastating for the plant. How does such a tiny parasite cause such a dramatic transformation?
Yeah, it's quite something. Even under the microscope, you can see individual root hairs swell up into these distinctive club shapes. And crucially, the infection turns the infected root into what scientists call a new carbon sink. This means the pathogen basically diverts all the plant's valuable sugars, the food made by photosynthesis for its own growth, directly to these swollen root clubs. It essentially starves the rest of the plant to feed the infection site.
That's a really clever, devious strategy. So how does the initial infection physically happen? You said it's injected.
Exactly. So when those clubbed roots eventually decay, they release masses of these spiny-walled resting spores back into the soil, millions of them. These spores are then stimulated to germinate by specific chemicals, things like ally isothiocyanates, which naturally leak from the roots of brassica plants. So the plant roots are basically inadvertently sending out a signal saying, come and get me. The primary zoospore, with its two little flagella, swims towards the root, latches on, and then forms that cyst we mentioned. And here's where the truly amazing microscopic engineering comes in, the penetration mechanism. The zoospore cyst develops this incredibly specialized injection apparatus. It really is like a miniature syringe or maybe a tiny gun. It has a long tube called a roar with a sharp bullet-shaped stylet inside called a stachel. Think of it as a tiny projectile. There's also this sticky pad, an adhesorium, that glues the cyst firmly to the root hair wall. Then a large internal vacuole inside the cyst rapidly expands. This creates immense turgor pressure, huge pressure. And this pressure is what literally thrusts the stylet through the tough plant cell wall. It's a rapid, explosive injection. And what gets injected? A tiny wall-less amoeba shot directly into the host cell cytoplasm. The plant tries to respond, forming a little callus plug around the wound, but the damage is done. This precise, forceful injection is really a hallmark of many Plasmodiophorales.
So wait, you're saying these microscopic organisms have evolved a built-in high-pressure hypodermic needle system? That is genuinely astonishing.
Absolutely. It's incredible microengineering. And once inside these plasmodia, they don't just sit passively. They actively manipulate the host. As the pathogen grows inside the cells, it messes with the plant's hormone balance. Infected roots show significantly higher levels of plant growth hormones, particularly auxins and cytokinins. The plasmodia themselves can even synthesize some cytokinins, like zeatin. For auxins, it seems they interfere with complex host pathways. Essentially, they hijack the plant's own growth regulation systems to promote this uncontrolled cell division and enlargement, leading to those characteristic club-shaped swellings. And the infection doesn't just stay in the root hairs, it spreads deeper into the root cortex, even the vascular tissues, causing really extensive damage and deformation. Eventually, these plasmodia mature and transform into masses of new haploid resting spores, ready to be released back into the soil when the root decays, starting the whole cycle over again.
Haptoglossa, the nematode nemesis and its gun cell. Okay, so we've seen how devastating these Plasmodiophorales can be to plants. But you mentioned some relatives, like Haptoglossa, target something completely different. Tiny animals like nematodes and rotifers.
Yeah.
And their infection strategy is just as amazing.
It is, yes, but with really fascinating similarities in the way they infect. These are also microscopic organisms. You find them in soil or maybe animal dung. Now, unlike Plasmodiophora, their main body, the vegetative thallus, is usually surrounded by a cell wall inside the host. And these thallus can grow to practically fill the entire body cavity of a nematode. Then they convert into sporangia, structures that release the spores. Some Haptoglossa release zoospores, like Plasmodiophora, but they're often weak swimmers and tend to insist quickly near the dead host. But other species do something even more striking. They produce non-motile spores called planospores. And these are released by an actual explosive rupture of an exit tube from the sporangium.
An explosive rupture. Okay, this sounds even more dramatic. How do these non-moving spores possibly find and infect a nematode then?
Well, this is where the gun cell comes in. These cysts, or planospores, they germinate to produce this elongated, often tongue-shaped cell. It's famously known as the gun cell, or infection cell, and it is truly one of the most remarkable structures in this whole group, maybe in all of microbiology. You need to picture a tiny, walled cell that functions just like a sophisticated miniature firearm. Seriously. It has a tube leading to a pointed tip. The opening, the muzzle, is sealed by a plug. Inside, there's a bore and a needle chamber containing the projectile, a needle. It's similar to the stylet of Plasmodiophora, but often even finer, more pointed, adapted for puncturing the tough outer skin, the cuticle of a nematode. The whole formation of the cell is incredibly intricate, involving complex internal growth processes, all while building up enormous internal pressure, turgor pressure, mainly from a large vacuole.
Okay, so we have this intricate biological gun. How does it actually fire at a passing nematode? This is wild.
Right. So, shortly before it's ready to discharge, that increasing internal pressure pushes the tip forward a bit, the muzzle plug is lost, and the tip forms this sort of beak-like projection. And this beak is coated with an adhesive, a biological glue. This firmly sticks the gun cell onto the cuticle of any unlucky nematode or rotifer that brushes past. This firm attachment is absolutely critical. It provides the resistance needed for what happens next, the explosive penetration. Once it's triggered, and the trigger might be chemical or maybe mechanical pressure involving calcium ions, perhaps structures holding the needle projectile in place rupture, and the immense turgor pressure of the gun cell immediately fires that needle forward. But what's even more astonishing is that the entire tube then explosively averts it, literally turns itself inside out, like popping a syringe plunger, injecting the cell's nucleus, Golgi, mitochondria, everything vital into the nematode, and the material that was coiled at the base of the gun cell. That forms the wall of the newly injected infective cell, now called a sporidium, inside the host.
That's absolutely mind-boggling. A self-propelling, adhesive, high-pressure, internal aversion syringe that injects a living cell payload. This just highlights the incredible, almost alien-like mechanisms that have evolved at the microscopic level. Truly amazing. Battling back. Control strategies.
Yeah, it's pretty stunning biology. But despite understanding these really intricate life cycles and infection mechanisms, actually controlling the diseases caused by Plasmodiophorales is exceptionally difficult.
Right. What makes them so tough to get rid of, especially for farmers trying to grow crops?
Well, the number one reason is the sheer longevity of those resting spores. Like we said, they can stay viable in the soil for up to 20 years. 20 years. That just makes short-term crop rotation, planting something different for a few years, almost completely ineffective against something like clubroot. Plus, the fact that Plasmodiophora brassicae can infect common weeds in the cabbage family and even some hosts outside that family, well, even if those weeds don't show the big clubby roots, they can still support the pathogen's asexual cycle. They act as hidden reservoirs, keeping the disease present in the environment even when the main crop isn't being grown.
Okay, so they're incredibly persistent. What measures do we actually have to fight back against these tenacious parasites?
Some general measures can help a bit. Things like improving soil drainage because the zoospores need water to swim, and applying lime to raise the soil pH, that can retard the initial infection of root hairs. But liming isn't a permanent fix. It might just delay spore germination, maybe even prolonging how long they survive overall. More recently, adding boron to the soil, especially along with a high pH, has shown some promise in suppressing infections. And it's absolutely critical to raise seedlings in clean soil, either non-infected soil or maybe steam-sterilized soil, before you transplant them out into the field. Oh, and interestingly, because some resting spores can actually survive passing through an animal's digestive system, you should avoid using manure from animals that might have eaten diseased plant material.
Hmm, okay. What about chemical options? Are there any effective fungicides?
Yeah, that's a tough one. Unfortunately, many compounds that used to be effective, like those containing mercury or the fungicide benomol, they're now banned in many countries, due to environmental and health concerns. So right now, there isn't really an economically viable and ecologically acceptable fungicide widely available specifically for clubroot, though research is always ongoing.
And biological controls. Any natural enemies we could use, like beneficial microbes?
People have certainly tried. There's been research into potential biological controls, but their full commercial viability, at least in the near future, seems doubtful. It's complex. Really, the most promising long-term strategy for clubroot, increasingly, is focusing on breeding resistant varieties cultivars of the crop plants themselves. For example, scientists have studied the common weed Arabidopsis thaliana extensively. It gets the full range of clubroot symptoms, and it's easy to work with using molecular biology tools. And they found that natural resistance in Arabidopsis can be based on a single gene. It involves something called a hypersensitive response, where the infected plant cells basically commit suicide very quickly, dying off before the pathogen can multiply. So you can potentially enhance susceptible crop varieties through genetic transformation, giving them these resistance genes.
Genetic resistance. That sounds really promising. Is that like a silver bullet for all the affected crops?
Well, not entirely, unfortunately. It's more complicated in some major crops. In cabbage, for instance, natural resistance seems to be multigenic, meaning it involves multiple genes working together, and it doesn't show that same clear, rapid, hypersensitive response. So breeding for resistance in cabbage is much more difficult. And even when successful, it might not last. P. brassicae is notorious for rapidly evolving new virulent races or strains that can overcome the plant's resistance after just a few years of that resistant cultivar being grown in a field. I mean, by 1975, they'd already identified 34 different physiological races in Europe alone. It's an ongoing arms race. Plus, even in resistant cultivars, the pathogen can sometimes still infect the root hairs and reproduce asexually via those zoosporangia. So it might still hang around as a hidden reservoir, ready to potentially evolve and break the resistance later.
So it's a constant battle. What about the other diseases you mentioned, like powdery scab on potatoes or cookroot and watercress? Are they equally hard to manage?
Powdery scab of potatoes, thankfully, is generally considered less economically important than clubroot. Good drainage helps manage it. However, the virus it carries, the potato mop-top virus vectored by Spongospora subterranea, that can be serious. But here, biotechnology actually offers a really promising solution. Scientists have created transgenic potato plants engineered to contain the gene for the virus's coat protein, and these engineered plants have been shown to be completely resistant to infection by the virus itself. So that's a big win. And for crookroot of watercress, control is achievable. It usually involves carefully adding zinc to the water supply where the watercress is grown, typically by dripping in zinc sulfate solution or sometimes adding finely powdered glass that contains zinc oxide, which slowly releases zinc into the water and inhibits the pathogen's infection process.
Outro. Wow, what a journey into this unseen world right under our feet. We've untangled the identity crisis of the Plasmodiophoromycota, learned about their incredibly destructive impact on crops we rely on, and marveled at their really intricate, almost science fiction-like infection mechanisms, especially that absolutely astonishing gun cell of Haptoglossa.
Yeah, this deep dive really highlights just how profound the impact of these microscopic organisms can be, from causing these massive swellings on plant roots to acting as vectors for devastating viruses. Their biological sophistication, things like those cruciform nuclear divisions, the complex life cycles, the sophisticated injection systems, it's genuinely remarkable for organisms often thought of as primitive.
So what does this all mean for you listening?
Well, I think it's a powerful reminder that even the smallest, most seemingly simple organisms have evolved these incredibly complex and, frankly, effective strategies for survival and reproduction. And it does raise an important question, doesn't it? Despite all our scientific advancements, all our understanding, why do these fascinating protozoan parasites continue to pose such a significant, persistent challenge, both to agriculture and maybe even to our fundamental understanding of life's adaptability? Definitely something to mull over the next time you're walking through a garden, or maybe even just looking at a potato in the supermarket. Thank you so much for joining us on this deep dive.
Yes, it's been a real pleasure exploring this fascinating microscopic world with you.
From the Deep Dive team, we'll see you next time.